So, hello, good evening, everyone. My name is Seemanti Aditya, as you all know by now. So, I am a Ph.D. student in Department of Biotechnology at IIT Madras, and I'm the teaching assistant for this course, Immunology, in this semester. So, today we'll be discussing on the week 12 content. So, it's about the vaccines and the passive and active immunization, and what kind of response the immune system has according to like different infection, bacterial or viral. So we'll just brush up on that a little bit. So before that, we'll see that so infectious diseases, so we know that a lot of infectious diseases are there in the surroundings, and a lot of people are dying from infectious diseases. So infectious diseases include all those diseases which are easily it could be spread in the population. For example, the largest example was the COVID-19 infection in the last 2 years, that has that the pandemic had caused a havoc, and it killed so many people, millions and billions of people. So apart from that, there are many other infectious diseases like the respiratory infections, the diarrheal diseases, HIV, tuberculosis, and malaria, etc. So all these infectious diseases, they count for around 11.2 million deaths attributable. This was on the basis of the 2008 WHO records. Now it is much more, and like after the pandemic, it has increased a lot, the graph, if you would see. So apart from that, there are other diseases as well which are causing death, for example, injuries and asthma, and neoplastic diseases or prenatal diseases. So all of these diseases we do not have a lot of control on, because a lot of diseases are like we do not in the point of immunology way. So if this kind of diseases can be not prevented, but it can be cured. But infectious diseases, there is a chance, or there is a possibility that we can prevent it, and we can prevent the spread of the disease. So there are a lot of vaccines that are coming up to inhibit all this disease spread, and the disease to be stopped at a particular stage, and not to take it into a pandemic scale. So the biggest example was the COVID vaccine. That was a very, very nice and very successful vaccine that came up, and that protected us, and we are now returning to the normal conditions. So so how does immune system respond to different viral infections? So the viral infections, they are actually causing the infection inside our body, inside the cells of our body. So a lot of time what happens is they can escape the immune system, because it can mask itself by being its own, by being in the own body cell, and so the body cell cannot so the immune cell cannot recognize it as foreign. So but instead of that, whenever, even if the body cell is inside the body cell, there is virus particles. So it will be at some point of time, the virus proteins will be expressed on the top of the cell, and these proteins will be different from that proteins which are normally present on the on the surface of the other normal cells. So the virus infected proteins, the virus infected cells will show different kind of proteins on their surface, and these proteins can be these proteins will be targeted by the antibody, and this antibody response and the complement response will be activated against the different protein molecules that are expressed on the surface of virus infected cells. So this accounts for the humoral branch of the immune system, and here we can see that there are different activities of different kinds of antibody. So antibody, the secretory IgA type of antibody, it primarily blocks the binding of virus to host cells. Thus it prevents infection or reinfection. So it inhibits the virus to enter the host cell in the first place. So in immunoglobulin A in its secretory form does that. Other antibodies, like types of antibodies like IgG, IgM, and IgA, it can block the fusion of viral envelope and host cell plasma membrane. So during virus entry process, there are two steps where basically the viral entry process has two steps, and that is first step belongs to the virus recognizes the host cell receptor. So this is the first step, and the second step is when the viral and the host membrane fuse together. So this fusion results in the release of virus particles into the host, and then the infection continues. So there are so the first IgA secretory type helps in blocking the first step of virus entry, and the other types of antibodies blocks the second step of viral entry, that is it blocks the fusion of host and virus membrane. And also there are other activities, like it will enhance phagocytosis of the virus particles, like the opsonization, or it can agglutinate the virus particle. It will neutralize the virus particle, and it will render it inactive. And complement activation also, complements they work by opsonization and by creating a membrane attack complex, or MAC, that produces a pore on the surface of the infected cells, and then cause the lysis of the cell. So this is the humoral branch, where the complements and the antibodies play a major role for providing protection against viral infection. In the cell-mediated branch, we'll have the T cell and the B cells, as well as the NK cells, macrophages. So T cells, they have the property, the cytotoxic T cells has the property for killing the virus. It has cytotoxic effects. It will help in creating a pore, and help in increasing the reactive oxygen species and all of that, and then it will kill those cells by creating a toxic effect. Also, it will secrete some kind of interferons. So these interferons we had talked about. These are antiviral and antiviral kinds of chemokines or cytokines, and they help in protecting the virus, and not the cells which are not infected with virus. They help in signaling those cells to protect it from further viral attack, and it has antiviral activity. Also, the NK cells and macrophages, they have the antibody dependent cells mediated cytotoxicity. This means, again, the antibody binds to the cell, and then that antibody finally causes the cell death, or apoptosis of the cell. So this is how the different branches of immune system is acting on to provide protection against viral infections. So so here I wanted to discuss about two very important thing about viruses, or say influenza virus in particular. So we know that influenza has a lot of subtypes. So here I'm going a little into the virology part. So influenza has a lot of subtypes. We'll see that H1N1 or H2N2. So what are these subtypes basically? So the subtypes generally corresponds to two proteins of the virus, influenza virus. H stands for the hemagglutinin virus protein, and N stands for neuraminidase. So these are abbreviated as HA and NA. And what happens is in the influenza virus, so this virus can change its, so this is the human host cell, sorry. So this is the virus, and the virus has the two surface proteins, namely the hemagglutinin and the neuraminidase. So these proteins, they can mutate itself, and they can change its epitope or antigenic property every year. So designing vaccines against influenza virus is a challenge, which could be universally applicable for all of the time. So influenza vaccines are always mostly annually given, because every year annually administered vaccines, because every year the influenza virus changes its epitopic antigenic properties. So your body does not recognize the previous year's influenza subtype as the memory cells are generated against the earlier pathogenic or the earlier antigenic property of this influenza virus. So every year what happens is, so this is the virus. It is affecting the person A in year 1, and due to some kind of point mutations which are arising in the genes of hemagglutinin and neuraminidase, there are certain point mutations which arises due to the activity of error prone RNA polymerase in the influenza. So influenza is an RNA, it's an RNA virus. So what happens is, when the RNA polymerase is synthesizing RNA in influenza, so there is some kind of, because it's very error prone RNA polymerase, so it shows some kind of mutations which are inserted or deleted in the genes of hemagglutinin and neuraminidase, as a result of which the properties of these proteins changes might be minutely, not that great, not that vast or diverse change, but very minute change. So you will see that the colors are different. Over here, this is light pink color, then it comes up to dark pink color and dark blue color, and then maroon and very, very dark blue color. So this changes its property over the period of years. So this is very small change, and this is known as antigenic drift. So it is drifting. So basically it is drifting from one set of protein sequence to another set of protein sequence, and this is antigenic drift. So due to this, although the person might recognize this antigen, but the immune response will not be a secondary immune response. It will be a very minute, it will again start from the primary immune responses. And next, what is more dangerous is what happened mostly in COVID, that is anticipated, is antigenic shift. So antigenic shift happens when there is an emergence of new subtype of influenza or a virus. The example is given here for influenza. So there is a new subtype of virus or an influenza that is generated because of mixing of genes from influenza virus affecting different species. Say influenza virus of chicken, and influenza virus which is affecting chicken, and influenza virus which is affecting human. So these two have different properties. So if these two viruses are somehow coming in contact to each other, then there will be a lot of change in their hemagglutinin and neuraminidase sequences, as a result of which, here as it is given, human influenza and swine influenza, so they are affecting the same an intermediate host cell. So this intermediate host cell here, this virus will, both viruses, they will come in close contact with each other, and they will show exchange of their genetic material, and this exchange of genetic material will lead to a development of a more greater very dangerous variety of the virus, and this property is known as antigenic shift. And this does not happen over the years. It happens like it's not like very prominent like antigenic drift, because this happens very commonly, very frequently, we know that. And but antigenic shift is very dangerous. It happens after a long period of time, after there has been a lot of mutation in these viruses and a lot of genetic mixing. So after that, this virus will get mutated, and then this antigenic shift is taking place. And this is very dangerous because this virus has the properties of both the different species of the virus, and so if it is affecting a human cell, it could be very deleterious and very difficult to generate an immune response against this virus. Also, there is something which is known as antigenic sin. So what happens in antigenic sin is, so there is a virus. This happens mainly in viral responses, because whenever there is a pathogen, so or a viral antigen, so this is basically recognized by a B cell, and B cell gets activated, and B cell produces antibody against this antigen, and this pathogen is eradicated. Now, during secondary response, if the pathogen retains its properties, like if the influenza has not changed its subtype, it is H1N1 is still H1N1 and has not converted to H2N1 or vice versa. So that time the pathogen is retaining its epitopes, so the memory B cell will recognize the pathogen, and the secondary immune response will be activated, antibody will be produced, and pathogen will be eradicated. But if the pathogen changes its properties, say the H1N1 gets converted to H1N2. So here what happens is your immune system will not be able to recognize this pathogen because of this minute change. Okay, because of this minute change, it will not be able to recognize the pathogen, and on top of that, it will again send a negative signal. It will send a negative signal to those B cells which are even trying to recognize this pathogen, like the new B cells which are trying to recognize the pathogen, the naive B cell which is trying to recognize the pathogen. So it will send a negative signal to those naive B cells that do not produce an antibody against this pathogen, because they are not sure enough that whether antibody is already produced against it, or they need to produce a new antibody. So they get like a little confused that this is pathogen which is changing its face and coming. So whether or not to produce a new antibody against it, or already our body has an antibody and we do not want to waste any resources. So this property is known as antigenic sin, which the B cells commit, where the primary response against this unique epitopes of pathogens are inhibited. So inhibition of primary response against unique epitopes on pathogens are inhibited, that elicit memory cell response is called original antigenic sin. And this is the reason why like over the period of time, if your viruses change their different subtypes, so the secondary infection, the secondary response, or the secondary immune response, or the memory response that we call it, continuously decreases. So your viral strain has, say, A, B, C epitopes. So the first primary response is very strong, and the specificity is very strong. As soon as the virus changes its subtype, say the C becomes D, so the secondary or memory response lowers, because the specificity is only against A and B, and D is not recognized. And as soon as there are two epitopes which are changed, only A is been recognized. So here your memory responses becomes very, very low, as well as this pathogen, when it is binding, this new epitopes, when it is binding to the new B cells, those B cells are also not getting activated or not getting proliferated, because there is a negative signal that is sent by this pathogenic by this pathogen, that by this, not this pathogen, but they get confused whether or not this pathogen is new, because they recognize one of the epitope but not the other two. So that way they commit a crime, and that is known as an antigenic sin. So only when the entire epitopes change, only when the pathogen totally changes its epitopes and comes as a new kind of virus, or new pathogen, so that time your primary response is again started. Otherwise your primary response is again inhibited. So this is something very interesting that I found. So that was about viruses and how immune system deal with different viruses. So how extracellular bacterial infections are dealt with. So this we have studied in our previous classes to some extent, like most of the thing we have studied. So how viruses are viruses, basically they are engulfed in your own cells, and so humoral and cell mediated immune responses are coming into play. But bacteria, mostly they are they are extracellular, and so the B cells, or the macrophages and dendritic cells, they become very, very accessible to them, and they can easily find them and kill them. So there are 4 ways how they are killed. So toxin neutralization, basically all the bacteria that are producing this toxins. These toxins can bind to antibodies, and these antibodies are secreted antibodies. So this secreted antibodies neutralizes the bacterial toxin. So these antibodies are also acting as an antigen apart from the bacterial cell itself. Then complement mediated lysis. So the complement proteins will go and bind to the surface of the bacterial cells. So here you see the complement protein is binding, and this complement protein will form a membrane attack complex, or MAC, and then cause lysis of this bacterial cell. Also it can, this complement proteins are binding to the cell. It is also allowing the antibodies and the other macrophages and other cells, phagocytic cell, to come closer to each other and helps in engulfing these cells, because complement proteins also act as an opsonin. So complements are opsonin proteins. So these are opsonin as well. And also they act as an anaphylotoxin, which mediate the mast cell degranulation. Basically all the A part of the complement proteins that are not directly associated with the activation or the lysis of the cell, this type of this subunits of complement protein, they act as the inflammatory signal inducer. So this act in inflammation. And also degranulation of mast cells. So degranulation of mast cell will release all of these enzymes and other proteins that will finally mediate anaphylaxis or a toxic shock or inflammatory response. So this is how a bacterial cell is tolerated by the immune system. So now, what is passive immunization, active immunization? Just briefly. So passive immunization, we know all the antibodies which are already prepared in another individual or another animal. So these are preformed antibodies prepared in another individual or another individual or an animal, that is administered or injected in a different individual to provide protection. So we are directly providing the antibodies, the preformed antibodies, to the individual against any kind of infection. So a great example is the antivenom that is given against snake bite. So the snake bite in horses, they can generate antivenom, they can generate the antibodies against snake bite. And it is not that toxic for them as it is toxic for us humans. So the snake bite, the venom of the snakes are injected in the horse, and then the antibodies are produced in the horse. So the antibodies are isolated from the horse serum, and then given it to the patients or individuals who have been bitten by a snake. So directly, because during that time the immune system does not have the time to produce antibodies against the snake bite. If given time, the antibody could be produced in humans as well, but there is no time to act against that very toxic venom of the snakes. So we need to give an immediate treatment to those patients, and that is why an antivenom is required. An active vaccination or an active immunization is, on the other hand, just administration of the pathogen itself on the in its inactive or its killed form to elicit an immune response in the individual. So the individual himself or herself produces antibodies against this pathogen. And so this provides protection against infection by the same pathogen in future. So anytime active immunization is much better than passive immunization, because the antibodies are produced in the body of the own individual, and there is very low chances that the next time if the pathogen is infecting, so there will be some kind of protection that will be given to that patient. But here, in case of passive immunization, that is not the case, because you need to continuously administer those antibodies again, because there are no memory cells against memory B cells or T cells which are generated against this against the venom, because you're directly giving just the secreted form of the antibodies. So active immunization is very important, and from there the concept of vaccines came into place. So vaccines are providing us protection against any future infection by generating the small primary immune response against the pathogen or the antigen against which we demand protection. So sometimes vaccines are not that sometimes the antigens which are used in the vaccines might not elicit an immune response, because they are too small in size, or maybe they are not that immunogenic and not recognized by the immune cells. So during that time, we need another substance which is known as adjuvant. An adjuvant is a molecule or a substance which will enhance the immune response when it is administered along with a less immunogenic antigen. So adjuvants basically help in increasing the immune response against an antigen, which in the absence of adjuvant would not have, triggered any immune response. So adjuvants example is like the mostly the aluminum compounds, the potash alum and the other aluminum compounds. Those are only the ones which have been, prescribed or suggested, and not any other adjuvants. Yet they are not prescribed to be used. So apart from that, we have some vaccines which are the recombinant and conjugate vaccine. So recombinant and conjugate vaccine, what does that mean? Basically, recombinant vaccine is like there are many categories of vaccines, like the killed vaccines or the attenuated vaccines. Attenuated means weakened, so the cells are weakened pathogens. Or we can have recombinant vaccine or conjugate vaccine or subunit vaccine. So these are different ways how scientists are still experimenting on what could be, injected in an individual to gather immune response against it. And so they have come across the vaccines like this. So different pathways by which you can generate vaccines. So killed pathogens and attenuated pathogens, this have been known for a long period of time, that okay, we can kill the pathogen and inject it, and then we'll get an immune response because the pathogen is no longer going to cause the infection, but they'll still have the epitope, and so the immune response will be generated against that epitope. Attenuated or weakened pathogens, this actually generates a greater immune response because it also activates the cell mediated immunity or the cytotoxic immunity. Kill pathogens, they do not activate the cell mediated branch. So attenuated vaccines are all attenuated or weakened pathogens. If they're administered, it is much better, but there could be one problem. The problem is that the weakened pathogens can somehow become activate, and then it can cause the actual infection. So that case, it could be dangerous, or it could be threatening to the patient. So here, in order to come across that limitation, the recombinant vaccine was discovered. So what does recombinant vaccine do is, so instead of the entire pathogen, so you are not taking the entire pathogen, rather you are taking only the gene which is responsible for encoding the particular epitope or antigen for the pathogen. Say the DNA which is encoding the antigen from this pathogen, you are isolating the DNA, and this DNA is then put into a plasmid vector. Here it is given the example of a vaccinia plasmid vector, vaccinia virus plasmid vector, and vaccinia virus plasmid vector has been widely used. So vaccinia virus is used for the the treatment against vaccinia virus. Was the vaccinia virus basically causes smallpox, and smallpox has already been eradicated, and it's a very weak virus. It is not having the so this plasmid does not have the capability to cause infection. So it's a very good plasmid to put your antigen and then generate immune response against your antigen without any infection. So this vaccinia virus has its own promoter, and it has a restriction cleavage site, and this is a gene which is a kinase gene. This is a viral gene as well. This is the vaccinia viral gene as well. So what happens is the DNA which you are isolating from the pathogen against which you want to generate an antibody response. So this DNA is incorporated inside this vector over in the restriction cut site. So your gene is over here, the vaccinia promoter is in front of it, and there is two flanking TK gene. Now what happens is, so this is the clone that you have made. This is the plasmid that you will, this is the recombinant plasmid that you want to inject and create and want to generate an immune response. So what will do is, so this plasmid is co-transfected with a vaccinia virus. So this is a vaccinia virus. It's a very weak virus. It's not going to cause any infection. Infection is not caused. So, the smallpox vaccine was the smallpox vaccine that was generated was against this vaccinia virus. So vaccinia virus, it's like a very it is now weakened virus and does not cause any kind of infection. So this virus is now transfected along with this recombinant plasmid in the tissue culture cells. So these are cells, so where you are transfecting both of this. Why we are transfecting both of this is because we have a TK gene. This is the viral gene, vaccinia gene. So TK gene is a viral vaccinia gene itself, vaccinia gene. So TK gene will have, so TK gene is also present in this virus. So TK gene is also present in this virus because it's a wild type virus. And so what happens when you're putting both of them together in the tissue culture cell? So this virus will replicate inside the cells. It is not going to cause any infection though. But along with its replication, the TK gene is also, having a kind of complementary part with this, the TK gene present in the vaccinia virus. So there will be homologous recombination of this plasmid and the vaccinia virus. Now you will have, so this virus will now contain this plasmid, like the plasmid containing this TK gene, and along with that also the pathogen, the gene from the pathogen against which we need a immune response. So this, because of the homologous recombination, we will get a vaccinia virus that is a vaccinia virus with the recombinant gene. So vaccinia virus, new vaccinia virus, mutant vaccinia virus is generated with the recombinant gene. So now in this vaccinia virus, you have this recombinant gene. So this recombinant vaccinia virus could be selected by BRDU selection. So BRDU is selecting only those vaccinia viruses which do not, which have this kind of recombinant gene. So recombinant gene, basically, they select on the basis of this TK gene properties. There is some process of selection how BRDU selects that we will not go into deep, but they select on the basis of the TK gene presence or absence of TK gene. And BRDU selection will only select those vaccinia viruses which are mutated and have the recombinant, this recombinant gene. So now you have isolated this recombinant vaccinia vector vaccine. So this vaccinia vector vaccine, so this is used as a this, if injected into your the host or injected in individuals, and this will generate an immune response, primary immune response against the virus, against the antigen which is encoded over here, whatever the antigen it's encoded. So this is a very good way of providing protection against the infection, providing the protection against whether or not the attenuated or weakened pathogen, because you're using whole pathogen over here in this criteria where this weakened pathogen is used. So there is a chance that it will get activated, but here in this recombinant proteins, it will not get activated. There is no chance of getting activated because you're not using the entire pathogen. You're using only a small part of the DNA which is encoding the antigenic part. So this is how the recombinant vaccine is generated. Another important vaccine that is generated is a conjugate vaccine. So conjugate vaccine is against, here the example is given against the Haemophilus influenza virus. So how this vaccine is great, a special one is they are conjugating the lipopolysaccharide or the sugar region of your antigen along with the toxin part. So both the sugar and the toxin is combined to make a conjugate vaccine. where they are linking the toxoid and the polysaccharide. The advantage for doing this is to activate advantages to activate both humoral and cell-mediated immune response. Because so your toxin is easily recognized by your antibody. So your toxin is recognized by your antibody, but your polysaccharide cannot be properly recognized by the antibody until and unless it is processed and presented by an MHC molecule. So the toxin's antibody is generated by the humoral immune response, but this polysaccharide gets processed and presented. So this gets processed and presented to the T cells by MHC, and so the T cells also come into play for providing protection. So this is how it is advantageous to totally link the toxin and the polysaccharide and prepare a vaccine. So another important thing over here is the immunotoxin that we might have heard if you have gone through the lectures. The immunotoxin is also again where the antibody is conjugated along with a toxin. So it's a very smart idea to do this because whenever you are trying to kill any cell, so this whatever cell you are trying to kill inside the patient, so this cell will have certain antigens. So this antigen will be presented on top of the cell, and antigen will be recognized by the antibody. So it will provide very, very specific response. So very specific response, because the antibody antigen interaction is said to be one of the very strongest interaction. And so antigen will be recognized by this antibody, and this cell basically, whichever is having your pathogen, it's mostly used for viruses or cancer. So antigen which is recognized by this antibody, and the toxin will basically go inside the cell and then mediate its toxicity and kill the cell. So it's how it could be used as a therapeutic purpose. So it's very smart idea where they are conjugating an antibody along with a toxin to provide protection against any kind of diseases. So that's all. Now we can go through some questions. So okay. Right. So the correct answer is option C. Formaldehyde is not used as an adjuvant because formaldehyde is a toxic substance, and it's generally used for fixing cells and not in kind of immune responses. So adjuvant is mostly the aluminium substances, potassium aluminium sulfate, aluminium sulfate, or aluminium hydroxide. So these are till now, these are only prescribed, and we cannot use any other kind of adjuvant to enhance the immune responses. So formaldehyde is the correct option. It's not an adjuvant. Correct. So all of you have given the right answer. So immunotoxin, as I just mentioned, so immunotoxin is where an antibody for a specific antigen is tagged with the toxin, and it could be used for um, so it could be used for therapeutic purposes. So that's right. The correct answer is option A. Right. So this was an easy question. So maternal antibodies, as we know that during childbirth, the mother passes the antibodies to the infant during the feeding, and so during that, the antibodies that are passed to the baby, so it's providing protection to the baby, and those naturally those antibodies are being passed, and we do not require any injection or do not require any kind of administration of those antibodies. So that is why that is known as a natural passive immunization. That's correct. Artificial passive immunization, antivenom, or any other way where you are naturally not receiving those immunization processes. So what's a conjugated vaccine? We use two things in a conjugated vaccine. Can just go through again over here. Conjugated vaccine has two things, basically a polysaccharide and a linked toxoid. So think again, I think, yeah, going in the right direction. No, so the answer is option B. It's the protein carrier, because see, here we had seen that the toxoid is used. Now, toxoids generally they are proteins, and proteins, they are recognized by the antibodies very easily. Polysaccharides, they are not recognized by the antibodies. They need to be processed. So the toxoid is linked with the polysaccharide again, as I said, to activate both the part of immune system. So the answer is not lipopolysaccharide carrier. So the carrier here is the protein only, because protein is carrying the polysaccharide inside the cell, where the polysaccharide is getting processed and where it is being presented. So B type of capsular polysaccharide will not be conjugated to another capsular polysaccharide. That is wrong. Lipopolysaccharide carrier, no. Polysaccharide needs to be carried by a protein only, not a polysaccharide carrier. So this is wrong. And a carbohydrate carrier also not. Basically, these are all same thing. Carbohydrate, lipopolysaccharide, and polysaccharide, these are all sugars. So we need a protein carrier to take the polysaccharide to the immune system and allow the polysaccharide to be recognized by the immune system and get activated. So the correct answer is option B, the protein carrier. And the last question for today. So for successful vaccine discovery, viruses are attenuated by, how are they weakened basically? So for, this is the correct answer is option A, not B. Most of you have given option B as the correct answer, because when you are passaging the live viruses from cell to cell, and then searching for the less virulent mutants using a low temperature. So low temperature means those mutants are well adapted to this low temperature, and they will basically be more weaker than those mutants which are adapted to higher temperature. One of the reasons is because this viruses, you are ultimately trying for a vaccine to be injected in humans, and humans have a body temperature of 37 degree. So if you are selecting these viruses from heat adapted mutants, then maybe there is a chance that they will prosper in your body, because your body temperature is
Ma'am, can this be one more that vaccines always require a temperature maintenance chain? So
right.
the option A is more correct, cold.
That is also, yeah, because they are already being produced in such a way that viruses are cold adapted only, like in order to, before injecting it in humans, they should be maintained at that temperature. So but another reason is that, like, after getting inside your body, so it does not get proliferated or does not cause the infection, just the immune response is generated. So yeah, right, that is also one of the reasons, like, why vaccines are needs a cold chain to be propagated or distributed. Yeah, right.
Thank you so much, ma'am. Thank you.
Yes, so this is the right answer. So thank you, everyone. So this was a very nice class and nice course to be a TA in. Uh, so basically, This was my first year duty in NPTEL, and I'm very glad that I had nice students. So before that, I'll just need to acknowledge one of my friends. So you might be seeing him like in most of the classes. So it's Prottay. So Prottay, can you turn on your video and say hi to everyone? So he has been like really great and being a great friend for conducting these classes very smoothly. And so yeah, so this was like a great experience, and thank you everyone. I would like that if everyone can switch on their videos for like maybe 5 minutes, and then maybe we can take a screenshot possible. I like to see everyone. So yeah, let me. That's great. I hope you learned, and I hope you had a nice time. I wish you very all the best for your future endeavors, and maybe again we can meet sometime if you have any
Thank you
doubt.
so much, ma'am. We are so happy to have you, ma'am.
Thank you so much. So
Share
yeah,
the same
everyone.
Thank you, ma'am.
that we are blessed to have such good TAs.
Thank you. I was really nervous. It was very stressful also, to be honest, because I had to brush up so many concepts regarding this. But yeah, it has really been nice, and you have been really good and
This
Yeah,
supportive.
so regular and also dedicated, ma'am.
arranged by IIT Kharagpur. How come the TAs are from IIT Madras?
Thank you, ma'am,
So
for your wonderful lecture.
welcome. So basically, actually, we need to do our tier requirements because of like the PMRF. So anybody can become a TA for an NPTEL course which is offered by another institute. So this was like because of that.
Do you need any eligibility criteria to be a TA?
Not as such, just like you. This is just our our requisite to be fulfilled. So for our PMRF TA duties, we need to fulfill these requisites or this criteria that we need to conduct.
PMRF, ma'am.
Huh?
Full form of that word you said
Oh,
PMRF or something.
PMRF is actually Prime Minister's Research Fellowship. So we are so we need to do that TA requirement like outside of our own college as well. So we need to be an TA inside the college as well as outside the college. So here, this is the TA requirement which I am fulfilling outside the college. So that is it. So thank you so much. You are great. We'll just take a screenshot. Everyone can switch on their videos for 5 minutes maybe and smile. Thank you, everyone.
Thank you very much, madam. Thank you.
Thank you.
Thank you,
Bye
madam.
bye. Have a nice and all
And
the
all the
best.
best for your future too.
Thank you so much. Thank you.
Ma'am,
Yeah,
ma'am,
Sashwata.
I have a question. Sashwat, ma'am, I have a question. Ma'am, I am currently doing MSc in biotech. In fourth sem, we have a project work. I can do it in your supervision.
What? I did not get you.
Ma'am, I am currently doing MSc in biotechnology.
Yeah.
Yes. So in fourth sem, you have a project work, like dissertation. Complete fourth
Yeah,
sem.
dissertations like you want to apply in IITs or like ISAs,
Yes,
or you
yes.
can. So for that, I think whichever professor you are interested to work with, you can just drop him a mail saying that you're interested in his work, and why you want to work with him, what particular area of the work you're interested to work. So you can just drop in a mail to any of the professor, and they'll reply surely. Like if you are very interested, just state them why what on work you would like to carry out in their lab, So you can mail them and then they will reply you back. So mail a lot of professors, because many of them might not see the mail and just ignore it. So, just mail a lot of professors, you will get replies from them, and then you can choose.
Madam, what would be the standards of coming exam, madam? Final exam.
Yeah, so the exam part, no, they will give you a mail. I think one mail has already been today. I was seeing one mail has already come regarding the exams and all. So what will be the standard? It will all, like, whatever is being taught in the class and whatever is there in the video lectures. So that is that will only be the standard, and already the questions that you have been solving in the assignments, and the questions that I have been discussing, so similar to that only. So that is what it will be. So don't worry about it, it will go good. All the best, you'll do great, don't worry.
Thank you very much, madam. Thank you very much.
Yeah. So then that is all.
Ma'am,
So yeah. Okay.
ma'am, can I
So
have some doubts? I have some doubts, ma'am.
yeah. You tell me.
Ma'am, ma'am, now what is the future of nanotechnology, ma'am? If we choose the specialization in nanotechnology, means it will help for us in future, and so I need about
Uh,
some
nanotechnology is like
some
I don't have a lot of idea
doubts
about it,
regarding
because
this nanotechnologies in medicine field, ma'am.
because see, this is like more towards the drug discovery, and so like nanotechnology will be used towards making good drugs in the future, and this cancer therapeutics, and a lot of like even the diabetic drugs as well. So to be like honest, I don't know that much about the drug discovery part. My area is mostly the imaging and microscopy and calcium channels. So see, the thing is, in biotechnology you need to it's not that industry oriented, to be honest. If you are very much interested in industry, so it's not that much industry oriented. But in academia there is a lot of scope. You can go on to do research, and then if you are lucky enough, you will get a patent for your nanoparticles that you might research and come up with. So that time you will be like in advantage, if you're talking about monetary advantage. So that is one thing. And otherwise academia is still great in biotechnology. Academia is good.
Madam, what is the fate of our biology students
Okay,
right now? Is it
thank you, ma'am,
and
for
in
the
IITs?
clarification.
Madam,
Welcome.
Madam, is that all seats are filling in IIT, madam, biotechnology or life sciences field?
Yeah, all the seats are getting filled. Like if you see, like during this PhD admissions or MSc admissions, nobody is basically leaving the seats. All the seats are filled. But like after certain, like maybe if they are not liking their research topic, or after admission, if they are not like happy with what they are working with, that time maybe they can leave it. But during admission, all the seats are almost filled. So as much as I know, because during my batch I took admission 2021. So what are the career options for immunology? So immunology is a like great way for research. Immunology, there is so much of research that is still pending, and lot of research is still not know, lot of things are not known. So academia is great in, like, if you want to pursue your education in immunology. Also industries are also taking people, like, for generating these vaccines, and all these industries, or the biotech industries, or the startups that are interested in the scientists working for them, for purifying the antibodies, for generating the vaccines. So that is also like a great thing. And like biology students, yeah, the seats are all filled up. But yeah, after admission, maybe some people, if they're not liking it, they might leave. But before, like during admission, there is no vacant seats.
Not for post graduation, for PhD, doctoral degree.
Yeah, PhD only, because PhD, see, they don't they don't give a number of how many seats are vacant. They will say that the admission cycles are open, but they will not say how many seats are vacant. But every year or every session, basically, there are 2 sessions of admission. So during the July session, there is almost 30 to 35 students on an average that they get selected. Now, I don't know what is the total amount of seat, because I think that 30 to 35 in every session is quite a good amount of people. In January session, I guess less will get selected, around 10 to 12 maybe.
Okay, madam. Thank you.
Yes.
Ma'am, I have a very simple doubt.
Yeah.
Ma'am, actually, my dissertation is in microbiology, and I want to, and I'm not willing to pursue it further in my PhD. I'll be appearing for interviews in upcoming months. Like I have recently cleared NET exam, so JRF exam. So most probably I'll be appearing for the interviews in upcoming sessions. In upcoming, so ma'am, will it be a hindrance to my admission? Like I have
No.
come. No, ma'am.
No, see, I also did my master's dissertation in a different topic, but my PhD topic is very different. It's just that what you have done in master's, you should know, because in interview they will ask you questions from your dissertation, and also like it is not mandatory that your dissertation and your PhD topic is same. People change topics like very.
Yes, ma'am.
Yeah, so don't get worried about that. That's not going to be a hindrance to your PhD admissions or your PhD, lab where you want to work in the future.
Okay, thank you, ma'am.
Yeah, welcome.
Ma'am, do you have any idea how click chemistry is used in biological field?
How chemistry is used? So chemistry
Click
is
chemistry.
click chemistry. No. The Nobel Prize that you're talking about. Yes. Yeah, I have no. I'm sorry, I don't know that.
Okay, thank you, ma'am.
Thank you.
Ma'am,
Yes. Rajkumar,
One
yes.
second, ma'am. Ma'am, the project any one topics, ma'am? Tell me, ma'am. New topics, immunology subject.
New topics regarding like what?
Yeah. Project.
Project
Anyone topics?
topics.
Yeah. Anyone question topics.
What could be project topics? So you can research on autoimmune disorders. I feel that is like a lot of research. They like how to cure
New topics, ma'am. Already one person.
Exactly, very new topics. I have no idea. No
New
clue,
are
sorry.
different, ma'am.
Anybody knows? Yeah, Jayappa, you want to say something?
It's better to go over choose immunotoxins if you want to different and new. Go for immunotoxin.
They're already known only. Like you will, whatever is already known, you can go and search. Like, see, whatever is known, everything is not known about that. So something is still unknown. Some link is still not known. So you can just go and just connect the dots, connect the link. So that is what you can do. Exactly, very new thing. If you want to discover, it is quite difficult actually.
During the journey,
And
maybe
also,
we discover.
every new discovery is basically based on what we have discovered previously. There is
Yeah.
always connecting the dots. We cannot just go beyond the literature. Like there is nothing on the literature, and we are discovering something new. That is
Yeah.
also not very
Yeah.
acceptable.
So that is it. So exactly, very new thing. It is not possible. Maybe after you have gone through your PhD and some few years of postdoc, then you will be having that idea that okay, maybe this can be done.
Maybe his journey, doing his journey, maybe he will invent something new.
Yeah, hopefully, that is what we are expected to do. Actually,
What happened on research only?
what?
That's what happened in research. Na?
Yeah, right. Yeah. So that is what. So we are basically connecting the dots. We are connecting what is known and what is not known, and then we are proceeding the journey. Yes.
May I know, madam, who is your supervisor, madam, research supervisor in IIT Madras?
Yeah, my supervisor is Professor Amal Kanti Bera.
Thank you.
Yeah.
And ma'am, can you please tell me what it means? How is the level of interview? Means what type of questions do they ask? Like, what is the do they ask your interest, like field of interest, or they completely go like whatever they want to ask, they ask something like.
So in PhD interviews, they will always ask what do you want to, what is your where you are comfortable in, whatever topic you are comfortable in. Say you are saying cell biology or biochemistry or something, so they will start you asking questions from those domains initially. So they will ask you something about signaling, something about this pathway, that pathway, GPCRs or something, and then slowly as you are answering the questions, they will basically take that question and then go into next question. So during that process, it goes into a like broader domain other than your cell biology as well as biochemistry. So whatever you are like initially, so get prepared about your dissertation. That is very important. Your dissertation topics, they will like what you have done, what you have maybe written in your thesis report. So they might be interested in knowing what you already know, and also your like basics. They will just brush up the basics and be like be cautious about answering anything, because whatever you're answering, so the next question is coming from that only. So and answer to the
Yes,
point.
ma'am.
Don't go into lot of description. If
Yes,
they're
ma'am.
asking what is this, just answer what is this. Don't go into like okay this that. Leave it for the next question to come.
Yes, ma'am.
That's why
Ma'am,
your
like
interest.
my core interest is in cell signaling. So will Lodish be enough for answering the questions, like, or do I have to go
Yeah.
through the current researches, like whatever is going on? So I have to have any idea of?
So wherever you are applying for the interviews, just see that what the professors are doing. You can just go through their papers, not everything, what they are doing and what they have, they know a little idea about. So that way you will not need to go into such a huge sea of literature. You don't need to search everything. Just
Yes,
go
ma'am.
through the papers of the professors in which institute like you are going for interview, and you are say you're interested in cell signaling. See which professors are performing those kind of research, and then go in their website and search about the papers that they have published, and go through that papers, and you will get like little link. Don't go into details, just what they have discovered, the little methods, what they have done.
Yes, ma'am. Thank you, ma'am. Actually, I'm a little nervous because this would be the first time I'm appearing for PhD
No,
interviews.
it will be fine. Don't worry. It's all right. It's all right. Be calm, stay
Thank
calm,
you.
and answer it like don't stress up. Even if you're messing it up, just like be cool and try to, even till the last question, you have a chance. So even if you are messing it up in the first few questions, don't get nervous. Be calm and answer the next questions that are coming. You don't know what others are giving answers, right? So they might be more
Yes,
poor than. So don't get nervous. Yeah.
yes, ma'am. Thank you, ma'am.
Nice learning journey along with you, ma'am.
Thank you, ma'am. It was very nice to be actually a part of this.
Thank
So,
you.
And
thank you.
for us also, like we do not have any connect with any scholar like studying in IITs. So this is the platform where we connect to the scholars they are studying there. So it gives us an idea. It gives us a clear
Right.
picture that what we have to go through in the next coming months. So it prepares us mentally, actually.
Correct. So, yeah, so don't worry actually. This is like a part of your journey. You will just learn along it. Don't take rejections personally, because if you're appearing, say you're appearing for so many interviews at a time, one interview did not go well, so don't get that into mind that okay, this interview did not go well, my next interview is also not going to go well. Don't think like that. whatever has gone, let it be, and then focus on the new part. So you will always, just be calm and just be positive. Everything will happen properly. Don't worry.
Yes, ma'am. Thank you.
Yeah. Thank you. I had a nice discussion with you all. So thank you, everyone. Bye bye.